We're learning today about nucleic acids from chapter 17, so we're getting close to the end of the semester. A nucleic acid, to define it, is one of our other big biomolecules, our last new one actually. We've seen carbohydrates, we've seen lipids, and we've seen proteins, so now we're looking at nucleic acids. And it's an unbranched, so unbranched just means it's one long strand, unbranched polymer composed of nucleotides. We'll see nucleotides, and there's a good list of definitions at the end of your notes here, so take advantage of those. But nucleotides are going to be a phosphate connected to a sugar connected to a base. And some of the colors that we're going to use for those, we will be using yellow today for our sugars. Bases we've been using blue for because they have nitrogen in them. And you will see that these high energy bonds between phosphate and sugar, those are called phospho-ester bonds, and those will be using orange. So we have a polymer of these 3-unit nucleotides repeating over and over and over again. So taking a look at nucleic acids, just to remind you, like, big picture where they are. So our nucleic acids are going to be found here in the nucleus. And if we take one chromosome, a chromosome is made up of nucleic acids coiled around histones, around proteins. If we take that chromosome and we unravel it, then we will be able to see the double helix of DNA with base pairs connecting in the middle. This is actually one of the X-ray diffraction pictures. So they take DNA and they make crystals out of it, which is very difficult to do, and then they shoot it with X-rays and you would get a picture like this. This is the type of picture that was used to figure out that there is a double helix structure to our DNA. So it's kind of the crisscrossing of the helixes that we're able to see in the X-ray diffraction here. There's some pretty cool stories surrounding, well, cool or bad stories surrounding those pictures and if they were stolen and things like that with Rosalind Franklin. But we're just going to look at the structure, the chemistry today. So in my long chromosome, there are small regions of it that are called genes. And those genes, sometimes you'll see them mapped where they're just individual lines here on the chromosome to show that there might be 100 or a couple hundred different genes all on a chromosome. A lot of the other space in between genes, it used to be called junk DNA, but it's not anymore. It's just non-coding DNA, but we're learning more all the time about maybe there's some use to that non-coding area there. So we have a gene, and then in between our genes, we have non-coding DNA. So just a couple of fun numbers with DNA. It actually was only within the last about 50 to 70 years that we've understood the structure of DNA. Initially, when scientists found these bases, they thought, okay, well, that can't be what builds a human or builds a plant. It can't be that simple. And it took them a while to figure out that it actually was that simple, that A, T, C, and G, which stand for 4 different bases, is all it takes to write the instruction book for building things. And that didn't make sense at first until they worked into it more. So in a human, it does take a lot of those base pairs to make a human. It takes about 3 billion base pairs. And when we talk about those base pairs, what we're looking at for base pairs is going to be these A's with T's or C's with G's. And you can see them here blown up a little bit. So A's with T's, C's with G's. On this space filling model here, those base pairs will be in the center of this molecule. And then you can see those base pairs also in the center of this double helix. If I see those written another way, just to give you yet another way you'll see those, sometimes you'll see them written like this, TGCA, and we'll see more of this 5' to 3' later, but my strands of DNA complement each other, A, C, G, T. And so here again are those base pairs. A's always pair with T's, C's always pair with G's. And across the middle there, what's holding these together is actually just intermolecular forces. So I'm going to do purple here and say those are my intermolecular forces. And you can see them here. So it's intermolecular forces that hold these base pairs together. Right down the middle here would be a bunch of intermolecular forces. That allows our DNA to not be permanently bound together, but it's Velcro or it's just kind of attraction. So I can open it and then I can zip it back together. And I can open it and I can zip it back together. And you need to open it to replicate, make a new DNA strand, and you also need to open it just to express that gene. So it's important that it's a low-key connection or a weak connection that you can easily separate, which is what intermolecular forces are good for. So back to the numbers here. We have about 3 billion base pairs to build me or you. In each gene, so in this smaller segment here, we would have between 20,000 and 2 million base pairs. And some of the research that's been done in the last few decades were to discover that not every single one of those base pairs actually correlates to something I'd see in a protein. Some of them are introns, some of them are called exons, and so there's a lot of editing that happens before you actually express that gene. Within a chromosome, one chromosome, you're going to find between hundreds and thousands of genes. In a human, we have about 20,000 protein coding genes. And that actually, of those genes, only about 1,200 base pairs are needed. So remember, there were 20,000 to 2 million base pairs per gene. But we only need a small fraction of those to make a protein. Protein's going to have around 400 amino acids. And we saw those proteins and amino acids back in chapter 16, right? So there's a lot of editing that goes on where I don't need all those base pairs to make a protein. Now here's something very cool. So this came out of the Human Genome Project, which was happening in the 90s. So they knew that there were about 100,000 proteins in our body. They were expecting to find about 100,000 genes in the Human Genome Project, and they didn't. They only found about 20,000, not because they did a bad job, but because it turned out that 20,000 genes are able to code for 100,000 different proteins. And again, that's going to be tied to editing, and that can be nature or nurture that turn on or off different expressions of those genes. So Project ENCODE is one of the projects that's going on now to figure out and map those proteins to the genes. And there's so much research that we're going to see in our lifetimes tying those genes and proteins and the expression of them and our health all together. So that's pretty cool stuff. So let's go back and just look at these pictures before we leave this page, kind of getting a big picture view of our nucleic acids here. We can see our base pairs here. If we go to this picture here, which shows us a little bit more of the chemistry, I'm going to highlight in yellow here some pentagons. And those pentagons are going to be my sugars. Notice they have an oxygen in the middle of them, which should remind you of chapter 14 when we saw sugars. And then we also are going to look at, with orange here, something called phospho ester bonds, or phosphodiester bonds. So phosphodiester bonds also. We'll see those in more detail. But basically we have sugars that are connected with phosphates in the backbone of my nucleic acid protein. And then some of them are only connected in one direction. So here's that nucleotide, phosphate sugar base. Here's a phosphate sugar base. There's a nucleotide. And it's that over and over and over again with just variation in the bases. And so if I connect that to this picture here, then what I would see in my backbone is I would see that I had a bunch of those phosphoester bonds, and then I have alternating that with some sugars. So they alternate there. Same thing over here, alternating sugars with those phosphoester bonds connecting them. And so I always have those nucleotides of a phosphate sugar base, orange, yellow, blue, orange, yellow, blue, orange, yellow, blue. And the blues are actually what are holding together and hold that genetic code, but they need the double helix to hold them in place. So that's an introduction to the big picture of nucleic acids. On the next page, we'll look a little bit more at the actual atoms and the chemical structures.